A high-level thought or plan can lead to action in the world by invoking the use of our muscles, or it can lead to subsequent thoughts. A high-level plan, say as high-level as a plan to go buy spinach at the supermarket that was realized in working memory after lots of deliberation about what one should make for dinner, is not only itself realized in neural activity, it can alter subsequent neural activity. That is, mind, comprised of various classes of thoughts and other informational structures such as plans, desires, hopes, and the contents of and operations within consciousness in general, can be causal in the universe. I have argued that mental causation is realized in a neural code where neurons alter the informational criteria for the subsequent firing of other neurons. This happens when those reset informational criteria have been met by inputs that meet those informational criteria at some point in the future. Before we begin an analysis of how a neural code could realize criterial causation, let's first review how neurons integrate information and fire. First, you can see here that neurons come in many shapes and sizes. The neuron shown here on the left is an example of a pyramidal cell. Pyramidal cells are called that because the cell body looks a bit like a pyramid or a cone. Pyramidal cells are the main workhorses of neural computation. They are always excitatory, so require other cells to inhibit them or to put the brake on their processing. These are the inhibitory interneurons that we just heard about in the last section. The anatomical differences among different types of neurons are not arbitrary, but rather belie differences in how neurons function, how they integrate information, and how they pass the informational results of their computations onto other neurons, whether locally to nearby neurons, say microns or tens of microns away. A micron or micrometer is a millionth of a meter, or onto neurons far away via long axons that carry signals called action potentials. These axons can be centimeters long, and in some extreme cases, like the long axons that feed down from the brain to the muscles, whether in us or in a giraffe or in a whale, axons can be meters long. For tiny nervous systems, axons would not be necessary. Neural computations could be carried out locally within local neural circuits in the absence of axons and action potentials, as happens in the local information processing that happens in our retina. Only when those computations are complete and it is necessary to ship the results of those computations far away are axons and action potentials necessary. In fact, there's a class of neurons in the retina called ganglion cells whose job it is to send the results of neural computations on to other parts of the brain via action potentials sent down their long axons, which bundle together to form our optic nerves. Thus, action potentials almost surely evolved so that signals could be sent to distant locations. Though they play a key role in neural computations, in that signals have to be sent from one neuron to another for further information processing, axons and action potentials are not necessary for neural computations, which can happen locally, as in our retina, in the absence of axons and action potentials. Even though many scientists have a bias to think of the neural code as realized in action potentials, we have to be aware that this is just a bias. For example, just because human information can be sent along telephone wires does not mean that the telephone wires are where the key information processing is taking place. Neurons never exist in isolation. They always exist in circuits of interacting neurons. For example, here's a drawing of the neural circuitry in the retina drawn over 100 years ago by the anatomist Ramon y Cajal, shown here sitting in front of his microscope. Because neurons process inputs in the context of circuits of neurons, it's almost certainly the case that information processing happens not only at the level of individual neurons, but also at the level of dynamic neural circuits. The correct level of analysis of information processing in the nervous system is the level of circuits. Some of these circuits might exist inside of a neuron, in particular in the logical operations possible among the branches of dendrites. And some of these circuits exist among interacting neurons. Given that the neural code is a neural computational code, it's likely that the neural code is realized in both dendritic computations within individual neurons and within the computations carried out among chains of neural computations, namely within neural circuits made up of many interacting neurons. That said, let's begin by looking at the organization of a single neuron. Here, you see a schematic drawing of a neuron. You can see a cell body here. You can see the dendrites of the neuron here, where the neuron takes inputs from other neurons. And here you can see the axon coming out of the cell body, which leads to the synapses of other neurons, typically landing on the dendrites of a downstream neuron, but in some cases even on the cell body of a downstream neuron. Here you can see the axon hillock, where an action potential is fired down the axon to other neurons that the axon terminates on. Every neuron has an input or dendritic end and an output or axonal end. This anatomy makes apparent that neurons take input from other neurons, process those inputs via computations realized within their dendrites, then send the results of those computations on to other neurons in the form of action potential signals. These downstream neurons take action potential outputs of earlier stage neurons as their inputs for further processing. Neural outputs, at least for neurons that are far apart, typically take the form of action potential signals, which are really patterns of electrical disturbance that race down the axon. The myelin sheath, shown here, increases the ability of axons to transmit these signals or action potentials to distant cells with little to no signal loss, a bit like a cable's insulation protects against signal loss. Here we see a depiction of a cortical pyramidal cell. Its cell body and its dendrites, both apical dendrites and basal dendrites, are colored red here, and its branching axon is colored in blue. The soma of a pyramidal cell is typically about 20 microns long. But if you include the dendrites, the length of a pyramidal cell can be hundreds of microns long, and if you include the axon, the length can get very long indeed. What we're especially interested in is what is going on at the synapse. The synapse is where the terminals of axons, or axonal boutons, of a presynaptic neuron terminate on a postsynaptic neuron. If we zoom in on a synapse, we can see that the synapse contains a small gap between the presynaptic neuron and the postsynaptic neuron. This is called the synaptic cleft. It's really tiny, on the order of 20 to 40 nanometers wide in most cases. By way of comparison, a human hair is between 80 and 100,000 nanometers wide. Since a nanometer is a billionth of a meter and a micron or micrometer is a millionth of a meter, the synaptic cleft is between 2 and 4% the width of a micron. Another way to put it is that the synaptic cleft is about 1/millionth of an inch wide. So the gap between the pre- and postsynaptic neuron is very small indeed, but it is a gap nonetheless. In a future section, we'll ask why there's such a space between neurons, but before we ask that, we should first learn what happens when an action potential arrives at the terminal bouton of a presynaptic neuron. In the standard case, when the electrical disturbance of an action potential reaches the end of an axon, or the axonal bouton, it triggers the release of neurotransmitter molecules into the synaptic cleft. You have probably heard about the main neurotransmitters. The main excitatory one is glutamate, and the main inhibitory one is called GABA. Then there are other neurotransmitters that modulate the degree or consequences of excitation and inhibition, including famous neurotransmitters like acetylcholine, dopamine, serotonin, and norepinephrine. We'll learn more about these classes of neurotransmitters and what they do later. The neurotransmitter molecules are held in tiny sacs called vesicles in the presynaptic neuron's terminal boutons. The arrival of an action potential triggers the binding of the vesicles to the presynaptic neural cell membrane, which then ejects the neurotransmitter molecules into the synaptic cleft, which is actually the outside of the presynaptic neuron. Once in the synaptic cleft, neurotransmitter molecules diffuse across the tiny space between the pre- and postsynaptic neurons. When the neurotransmitter molecules reach the postsynaptic neuron, they typically bind to a receptor that is specialized to take exactly this type of neurotransmitter as its binding input, much as a lock can only take keys of the right shape as inputs if the lock is to open. A receptor is a very complex little machine made of proteins that floats in the neuron's cell membrane, as shown here. Once the neurotransmitter has bound to the receptor might do something in response to that act of binding. In coming sections, we'll learn about different classes of receptors and what the consequences of neurotransmitter binding to them are for the firing behavior of neurons.